So in Messelson and Stahl's experiment to figure out how DNA is replicated, they did two things. And so if you'll just look on the left right now, what they did was they initially grew bacterial cells with nitrogen 15. It's a heavier isotope of nitrogen. When they grew that bacteria with nitrogen 15, that means that the DNA found in those cells were all labeled with this heavy nitrogen material. To determine that the DNA was actually heavy in nitrogen, they would actually put the DNA in what's known as a centrifuge, and that could actually help us determine the weight of the DNA. But all you need to know is that the original DNA was heavy in weight due to the nitrogen 15. After we had the original cell having its original DNA being heavy, labeled with the heavy isotope, we actually moved the bacteria to a different dish with nitrogen 14, a light version of the isotope, or a light isotope. So why does it matter that we labeled it with the heavy nitrogen, and then later on allowed these cells to move into this lighter medium? When they moved to this lighter medium, we actually let them replicate, allowed them to replicate 2 times. Okay? When we allowed them to replicate 2 times, there were some expectations as to what would have happened. We had 3 ideas for what happens when DNA gets replicated. The first idea is this conservative model. In the conservative model, we would expect that when the DNA replicated the first time, we would have one strand of DNA that was all light, made of this new nitrogen, and one strand that was all heavy, because we conserved or saved our original strand and used this as a pattern. In the second model, the dispersive replication, what we thought would happen is that this original molecule of DNA would basically break up into separate pieces and be split up or dispersed between our 2 new strands of DNA. So we'd actually have this isotope weight being medium, because it took in some of the light, the new DNA, the dark one here is the light weighted isotope, and some of that heavy. And so the overall weight during this first replication would have been medium. Same thing would have went for our third idea, which was the semi-conservative model, which says we didn't chop up this original strand, we just took one whole strand and put it into each of our new daughter strands. So we just broke the strands, separated the strands, and used one in each. So it was half old, half new, or having a medium weight, because they were equal parts, old and new. Then we allowed them, remember, to replicate 2 times. So when they replicated 2 times, in the second replication, those daughter DNA strands, we would have expected again to have some light strands, again to have some heavy strands. In the dispersive replication, we would have assumed that again we would have medium weighted strands, but we'd also have some light strands, and there would be equal amounts of these. Then, in the semi-conservative replication, we'd actually observe there to be more light strands. Because if you imagine, we're only taking one of these DNA strands. We're going to split the heavy and the light strand, and then we'd actually get more light. So if you imagine my blue being this light strand, okay, blue being the light strand, pink being this heavy strand, so this would be 1 and 2. So here's my 1 and my 2. And then I would be using, because I'm still growing it in this light medium, more light molecule. Okay? And so then we would have expected more light isotopes of the nitrogen 15. This semi-conservative replication ended up being what was true, what we actually saw. And so this proved the semi-conservative replication theory that each strand of a daughter DNA is half old and half new, okay, containing half of the genetic material. But moving on, so what actually happens with DNA replication? There's 4 main steps. The first is that we unzip the DNA. We do this by breaking the hydrogen bonds between the nitrogen bases. So right around here, we're breaking those hydrogen bonds. You can see them being separated. We will complementary base pair these molecules. So you can see right here between my 2's right here, I'm base pairing the nitrogen bonds left and right. Then I will be joining together the phosphate backbone, which would actually, since my black strand is the new strand, would be 3, is happening on both sides. And then I would eventually proofread with enzymes, which happens for all of these, and that would be a number 4 at the end. Okay? And again, my end result being that each new DNA molecule contains one old strand, or you can even call that a parent strand, and one new strand. Okay, this representing my old strand or my parent strand, and the black representing the new. All right, and so we have some enzymes that we want to mention. So this is a really complex scenario, so we'll actually talk about these enzymes tomorrow. Make sure you take some time to actually look at this picture right here. There's lots of new vocabulary that you'll want to look at, but try to see if you can figure out what's going on here, okay? And you might even want to look ahead and try to look at some of these vocabulary terms before we meet tomorrow.